Here we go. All right, so today we're going to pick up with the second part of what is now chapter 16, 16.2. Part 2. So still we're talking about the acquired response or the acquired immune response or the specific response. And like last time, we had talked about the whole humoral response, right? Pre-B cells coming from the bone marrow, being partially mature, getting their IgD on their surface, so they're specific for antigen, going out into the lymph, maybe in a lymph node, meeting antigen, being clonally selected to proliferate and differentiate, right, into memory B cells and plasma cells. Those plasma cells are going to release immunoglobulins. The immunoglobulins, we talked about that. Yes, the things that immunoglobulin can do for you and the 5 different categories of immunoglobulin. Yeah? Yes? Okay. So today I'd like to focus on what we used to call the cell-mediated response or the T cell response. And it starts the same. The immature cells are going to come from the bone marrow, but instead of going right to the lymph, they take a short little detour. Okay, so they're going to originate in the bone marrow, but then they are going to head to this organ, the thymus, that we talked about 2 or 3 lectures ago. It's really large when you're an infant and shrinks during your lifetime. And we say in the thymus, the T cells get educated, because that is really what T stands for. It stands for thymocyte. And what we really mean is they're being selected, and some of them are going to proliferate, and some of them are going to differentiate, and some of them are going to die. That's a word for programmed cell death, apoptosis, or sometimes you'll hear people say it apoptosis, right? And that means any cell, any T cell that comes into the thymus and its antigen binding site matches closely any of your self proteins, that T cell is going to be triggered to die, shrivel up, get removed, because that helps prevent an autoimmune disease. Right? So many autoimmune diseases have to do with when T cells that should have been eliminated get through the thymus and they go to wherever. Let's say it's multiple sclerosis. They go and they bind to the myelin that's coating your neurons. That would be bad for you, right? And they cause an immune response in the wrong place, right? So the thymus is really important for all of these things to go on. And the cells that come out of there, we're going to talk about 2. Many fewer cells come out of the thymus than went in, right? We used to say millions go in and hundreds of thousands come out. Or I used to say thousands of students come to Camden County College and hundreds graduate at the end, right? Same kind of thing. You get selected against for whatever reasons, right? And so the T cells that come out of here, the ones that we're going to focus on are going to be, as I told you guys before, cytotoxic T cells. Remember, we abbreviate those like that, T with a capital C subscript, and helper T cells. And there are lots of different kinds of helper T cells. We are not going to talk about all of those different subsets. All right, so now sadly, we're gonna have to go back and look at a picture from before. So remember this picture when we talked about class 1 and class 2 MHC on the surface of cells, right? Make it bigger, right? So all your cells have class 1 MHC on them, right? And remember, I'm going to draw it similarly when I draw on the board with those 3 bluish circles and a yellow circle. And your cells that are going to process antigen and present it to the T cells, they're also going to have class 2 MHC on the surface because T cells have class 2, right? Because they're part of the immune system. But they also have class 1 because they're part of you. We're trying to get you to understand that that surface of an immune cell, there's lots of different receptors on there and multiple copies of each of these receptors. It's a really busy place. And there's a lot of co-binding that has to go on to get a cell to do something. All right, so here's our friend, the B cell, that was up the whole time that I was talking on the last Zoom, unfortunately, right? And so we've got an antigen binding site here. We got like an IgD stuck in the surface of the B cell. And we also have class 2 because B cells are part of the immune system, right? What I want you to take a look at today, a little closer look at today, is the T cell, surface of the T cell, because that's kind of where we are right now, right? So we've got class 2 MHC on the T cells. We also have, see this green thing? That is, we call that the T cell receptor. And it's it looks like that, particularly in helper T cells, not as much in cytotoxic T cells, but it actually looks, and it kind of looks like an immunoglobulin. You know, it's got a couple chains of protein sticking out. It's got a place to bind antigen. These blue sticks are called CD3, and they are always on the surface of helper cells, of helper T cells. It helps us figure out that this cell is a T cell, not a B cell. And then embedded in some of those CD3 molecules is this thing that kind of looks like a hook or a paddle, and that's going to either be CD4 or CD8. CD4 on helper cells, CD8 on mostly on cytotoxic T cells, although we're going to draw it differently. Okay. Uh-oh. This is where I got in trouble last time. Okay, close that. There we go. So I'm going to draw on the board how a cytotoxic T cell gets activated first, because that one's a little simpler. And then we'll talk about how helper cells get activated. And let's see. I'll use my curveball. Okay, so when a cytotoxic T cell gets activated, the first part of this story is one of your cells, that's supposed to be the nucleus, a host cell that we refer to as a target cell, because it's going to be targeted for death. Right? And why this host cell is going to be targeted for death and not a different host cell is because this cell's been infected with a virus. So I'm going to draw sort of our standard animal virus, like that we looked at way back in chapter 6, right? And I'm going to remind you, this is going to come in and an endocytic vesicle, take off its coat, right? And cause this host cell to start making viral particles and viral nucleic acids. And I'm going to make some viral particles like these surface proteins here, right? And I'm trying to make them look like half circles. And then obviously there'll be some of the capsid, blah, 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 right? Some of these viral particles are going to get packaged up with some class 1 MHC, and they are going to be put on the surface of this cell. So remember, our class 1 has those sort of like those 3 blue circles in a row, and then a yellow circle, right? And now part of my antigen is going to be stuck to this. It might be at the end, it might be next to it like this. Different folks are going to draw it different ways. So we're going to have on the surface, because this is a host cell, this is me, right? This is like a liver cell or a skin cell, and it's infected with virus. And so while the virus is using my cell to make all its bits, sometimes some of those proteins are going to be put on the surface, we say in the context of class 1 MHC. So to trigger a cytotoxic T cell, the cytotoxic T cell needs to recognize, or we say C antigen in the context of self antigens, but in particular of class 1 MHC. So I'll put a T cell over here now, a cytotoxic T cell. I'm trying to make them a little smaller. Yesterday when I did this in my morning class, I made my cells so large I didn't have room to draw the helper cells over here. So I'm trying to moderate my space a little bit. So this is a cytotoxic T cell. And remember, it's a cytotoxic T cell because it's going to be filled, it's going to have some compartments that are filled with granules, enzymes, proteases, and we refer to these particles as perforins because they perforate the phospholipid bilayer, and granzymes because they are granular, like the granulocytes, but we don't want to call them histamine or cytokines because we've already kind of used those words to mean something else, right? And they're not really interleukins because this is going to kill one of your host cells, going to kill one of your cells, not another immune system cell. So we had to come up with a different word, okay? So these packets of perforins and granzymes, that's what's going to be the effect when the cytotoxic T cell recognizes antigen in the context of class 1 MHC. So, my glasses, so I can be sure to draw this correctly because it's not simple, that's for sure. So this is going to be our CD8 molecule here. And it is going to bind to the antigen. And your book draws it a slightly different. They draw it more like this shape, right? But it's bound to antigen and class 1 MHC. This is a cytotoxic T cell. It's still a T cell. It's going to have class 2 on its surface. It's going to have other types of class 1 on its surface, right? But this CD8 molecule is what's going to function as the T cell receptor, so to bind to antigen in the context of class 1 MHC. So I'll label all these parts, right? So this is class 1 MHC, and that's on the target cell. This is antigen, and I'll label it viral antigen on purpose, right? Since that virus antigen. And then this is the CD8 molecule. When this happens, and cytotoxic T cell needs to recognize or see antigen in the context of self. The CD8 molecule on the cytotoxic T cell is what binds to antigen plus class 1 MHC. Then something's going to happen. Then that cytotoxic T cell is going to release its granzymes and perforans. And those, so this vesicle will come over here, merge, right? Dump these granzymes and perforans out of the cytotoxic T cell, and they're going to go over here and they are going to kill this cell. Death. Kill the target cell. Lovely. That's going to make a big mess, right? Because we've got virus in there, we've got all the parts of a cell in there. And so what that's going to do is that counts as tissue damage, hence back to the other chapter. So that's going to trigger a little bit of inflammation. These molecules broken up, as well as these guys, are going to contribute to a chemotactic gradient. So macrophages are going to come here and they're going to be like, oh, do we need to clean some stuff up here? Which we're going to see when they do that, that's going to trigger that other part of the immune system, the other type of T helper cell, the other type of T cell, the T helper cell. This is nice and tidy because it's an actual effect, right? What we saw before with the immunoglobulin, it was just binding and it could sort of do some things, but mostly it enhanced phagocytosis, right? And clumped antigens together and occasionally blocked a virus or a toxin from actually binding to your cells, right? Questions about that before I start blithering on about the next thing? Yes, no, maybe so. Heard that story before? Those of you that have had AMP2? Yes. Yes. Of course. Just humor me. Just shake your heads yes. It'll make me feel better. Okay. So I am going to erase a little bit of this because I'm going to need the space. So binds to, I'll put that underneath here, binds to antigen plus class 1 RHC, causes granzymes and perforans to be released, which the effect is to kill the target cell. Not helpful. All right, so now what about the other part? What about those helper cells? Okay, they function a little bit differently in how they recognize antigen. They're going to need to see antigen or recognize antigen in the context of class 2 MHC. And there is going to be an actual T cell receptor that's going to bind to that antigen and the class 2 MHC. So T helper cells. So draw a picture, but how they get the bit of antigen in that context of class 2 is, so first I'm going to draw a macrophage, like a big phagocytic cell. Remember, that's the shorthand for macrophage, right? And I'm going to put my favorite bacteria over here, right? Put some DNA inside of it. And I'm going to try to put triangle antigens on the surface of this bacteria because I've already used circle for the virus, right? You all know I can only do triangle, circle, square. So this is triangle antigen. It's part of a bacteria from phagocytosis, from what we learned last week, right? This bacteria is going to get ingested, right? It's going to go into an endocytic vesicle that's going to become a phagocyte, right? Or phagosome. And the phagosome is going to fuse with the lysosome to become the phagolysosome, right? I'm going to put my bacteria in here dying a slow, painful death. So there's some of my bacteria, and here now they're all broken up into bits. Put some triangle antigens on there. So what happens is the triangle antigens survive the phagolysosome, and those antigens are going to get fused with another vesicle that is going to put those antigens on the surface of the phagocyte. So there's a little bit of a blow up of this on this picture. So get that out so that I can do it correctly. So let's put my class 2 MHC plus some antigen. So I think we drew class 2 MHC before. Remember, it kind of looked like a kidney thing. I don't know why I think that it kind of looks like a bean, right? But this one's going to have a binding site for triangle antigen in it because it's already going to be bound to triangle antigen like that. That is going to get put out on the surface of this cell. I'll try to stick it someplace where it'll be able to bind to a T cell. So here's my class 2 MHC. Right? This class 2 MHC. And the book draws it like that. And I'm going to just stick my antigen's going to get a little bit bigger now. I'm just going to stick my antigen in there. Antigen, right? Class 2 MHC plus antigen. And again, there's going to be multiples of these all around the surface of the cell, not just one. This cell, this macrophage, it can also be a B cell, or it can be a dendritic cell. Not usually a monocyte, not usually a neutrophil, not usually a neutrophil. Those are technically more involved in phagocytosis and then they die. These cells, we refer to all of them as APCs, and that stands for antigen processing cell. We used to call them antigen presenting cells. I think that makes a little more sense because it's presenting the antigen in the context of class 2 MHC to the helper cell. But people like processing to help us remember it's a phagocyte, right? So it got here because of phagocytosis, and now it's doing the phagocytosis, and it's putting a signal out on the surface. But we don't want to kill this macrophage because this macrophage has all kinds of other work to do, right? Remember, it has Fc receptors for immunoglobulin. So if some of these are covered with immunoglobulin, that's going to help bring more of them in, right? So there's all kinds of other stuff. The macrophage is trying to just get rid of the bacteria. So we don't want to get rid of it. So we're not going to kill the cell this time. We're just going to use it as a trigger to boost the immune response. Okay, so now I'm going to draw my T helper cell over here. I know it's a little bigger than the cytotoxic T cell, but not on purpose. T helper cell. Okay. And my T helper cell is going to have a T cell receptor on its surface, CD4 on its surface. So again, I want to draw this correctly. Don't want to mess up. So I've got my T cell receptor, which has those weird little wings on it that make it almost look like an immunoglobulin. It shares some peptide sequences in common with immunoglobulin. And this one, of course, is specific. This one's going to have a shape at the end of it to bind to triangles. Because I can't repeat this enough, that this is the specific immune response. So this helper cell isn't going to bind to circle antigen or square antigen. It's only going to bind to triangle antigen in the context of class 2 MHC. So this green thing, that's the TCR. I know, how many acronyms do we have to have? The T cell receptor, right? And then I've got some class 1 MHC, or sorry, class 2 MHC, but I don't have a different blue, so it's gonna have to be that same. So I've got that. I've got some CD3 molecules here. Those are going to be in blue. CD3. Those are the blue things. And as part of then that CD3, I guess I'll use red for that. Embedded in that, there's going to be some CD4 in this case because they're helper cells. So I'm going to put this, your book just kind of makes it look like a hook or a paddle, like that. And that red thing, the red thing is CD4. So we've got antigen, T cell receptor, and CD4. This is a co-binding, this is a required co-binding. And I know you guys on the Zoom won't be able to see this, but it is as if, right? I reach out, I'm shaking hands with the antigen presenting cell, right? And I'm like, oh, wait, I kind of know you. Oh, it's Jennifer, right? And this is my CD4 coming around to bind to the class 2 MHC. Go, oh, yeah, I do know you, right? Now I can go do my job because I know it's you, right? And I know your antigen, you're foreign, but I know you, right? Because my CD4 is binding to class 2 MHC, that's us. So I'm like, okay, this is a double bound confirmation. Once that happens, okay, so the T cell receptor and CD4, and I'll just put over here on the T helper cell, bind to antigen in the context of class 2 MHC, right? Because there's the class 2 MHC, and I got a little antigen sticking out of it. My T cell receptor binds to the antigen, like when I shook her hand, and then my CD4 comes around and says, oh yeah, you're class 2, us. We're all self here, great. Now I'm ready to do my job. When this happens, then that helper cell is going to release many cytokines. We're going to focus on interleukin 2. Okay. If this is a T helper cell type 2, it's going to release a little bit of interleukin 2 and a lot more interleukin 4 and 5. If it's a T regulatory cell, it's going to release a little bit of interleukin 2 and some other things like IL-17, right? So what actually the cell is going to spit out kind of is based on what its job is, what it was differentiated into, okay? But for our purposes, let's just try to keep it simple, right? So let's just focus on interleukin 2, because this is a really powerful interleukin in that the first thing this does, it used to be known by its other name, B cell growth and differentiation factor. So this is going to help B cells proliferate and differentiate. And we know that means plasma cells to make immunoglobulin, and our friend, the B memory cell for the next time. That's one thing it does. It really ramps up that B cell activation that we talked about last time. Another thing IL-2 can do for you is it is auto-stimulatory for T helper cells. So what that means is, I don't know what color I should make. So here's some endoplasmic reticulum, here's some Golgi, right? So here's a vesicle that's going to be filled with interleukin 2. A different color. Let's make it yellow. Oh, it's going to have to be squares. So this is this is your IL-2. When this IL-2 gets released, so some of it is going to go help the B cells. All right. Some is off to help B cells. Some of it is going to bind back to this very T helper cell. Oh, that's a nice color combination of purple and orange, right? So complements. And you're like, okay, so what? Well, we want more of these T cells. We want them to proliferate and differentiate as helper cells, because right now we're fighting antigen that's triangle. So we need more T cells that have a T cell receptor for triangle antigen. Okay. So the IL-2, we say it's autostimulatory because it's going to cause this T cell to undergo mitosis. And it can help other immature T cells become T cells like this that are specific for triangles. All right. But wait, there's more. All for the low, low price of Microbiology Community College. You can also learn the third function of IL-2. The third function of IL-2 is that it helps cytotoxic T cells too. So it's going to help cytotoxic T cells proliferate and differentiate, because we want memory cytotoxic T cells. And I forgot to say, here we're going to get also proliferation plus memory T helper cells that are specific for triangle antigen. So the next time I see this bacteria with triangles all over it, I'm ready, because now I've got hundreds of these T cells in my lymph circulating around looking for triangle antigen. All right. Questions, comments, worries, concerns? Yes, no, maybe so. Oh, wait, I'll move. Go ahead. If you want to get closer, that's fine. All right, now what? Cat, now what? Okay, now I want to show you some pictures, some other pictures. All right, so the next thing I want to have us spend some time. Are you guys finished photographing, or did I get in your mind? You're good? All right, so the next thing that I wanted to talk to you a little bit about was the amnestic response and how that works a little bit before we, here it is. I might have that in the PowerPoints. Let's just, let's share screens for a minute and see, because that would be good. And where did they go? Yeah, I think I have that picture. Let's make it big. You can't see that yet, right? Okay, sorry. I'm going to make it so just a way people could see it. Okay. So this is a picture of the primary and secondary response to antigens. And a lot of times I show this at the end when we talk about immunoglobulins, but sometimes, like for us last week, we were kind of about up to our eyeballs with what more information we could absorb, right? So I was like, I'll save that for next time. So it might be a little easier for you to wrap your heads around now as well, because the first line here, right here, this is your first exposure to antigen. So that means when you were bitten by the mosquito, or when you got the bacterial infection, or when you got the virus from your baby, your child, your spouse, your partner, right? So this first exposure, I need you to notice that we call this after the antigen exposure. There is a latent period. And if this is the first time you've seen that antigen, that can take up to like 12 days. This is like a 9 to 12 day period. Maybe if we push it 7 days. Where your body is doing all those things that we talked about, maturing some B cells, getting some T cells activated, getting those helper cells to help back to the B cells to make more immunoglobulin, to get enough immunoglobulin in your system that antibody can do all those things that we listed, right? And what I need you to notice here is the red line is IgM, the blue line is IgG, okay? So we talked about how IgM is always the first immunoglobulin, and you make a fair amount of it. This is in micrograms. Then notice it drops off pretty fast though, like 3 days, 5, 6 days, it's gone. And we get a nice IgG response. And the purple line is these two added together. How much total immunoglobulin did you produce? So first you make a little IgM and then a lot of IgG, and then you get better, right? Some time goes on and there's some sort of an interval here, right? So you get what we call a primary response. And that primary response is mostly IgM, then some IgG. And we think, oh, that's great, because the IgG lasts like 45 days in our serum. So it's going to be around a while to help us fight this infection, even after there's not that much of it left in our bodies. Your second exposure right, is going to cause a secondary response, which is also known as the amnestic response. And two things I need you to notice about that. Look how much more total immunoglobulin there is, a lot, almost double. And most of it's IgG this time. Right? We barely get any IgM and it's a little bit delayed, and we get tons and tons of IgG. So we get more total immunoglobulin, mostly this time IgG and a little bit of IgM. Now, why am I bothering to show you this? Because this is how vaccines work, right? This is what this is about, right? If you never get that primary response because you weren't vaccinated, then you have to wait this latent period to make a response. And it might be too long of a period. You might be dead by then. Or very, very ill with secondary infections, right? Which brings me to a little chit chat about vaccines. So let's switch this off. Stop sharing that. And I want to share this instead. I'm just waiting to see if it'll autofocus. Sorry. Sometimes no good. Please. There we go. Okay. So if you could see this whole diagram at one fell swoop, I'll try to make it small enough that we can see more of it. Right. This is table 16.6 in your book. And there are basically two different types of vaccines that we currently have. One of them is called a whole vaccines or whole cell vaccines. And your textbook did have a really nice breakdown of that I want to include. Right. So one type is whole cell, meaning whole bacteria, or whole virus. Okay. And there are subcategories here. They can be what we call live attenuated. That means they are weakened and they can't actually cause an infection, but there's enough of them, enough of the whole thing intact to give you a good primary immune response. Right? How do they attenuate them? Usually with heat. Occasionally with a chemical, but usually with heat. Another type of whole cell is killed. Killed cells, like killed bacteria, the whole thing though, or an inactivated virus. So a virus that's not able to reproduce, multiply, right? Viruses are not living things. I have to mute that or they're not going to be able to see. That's great. There are also some that we use, we say a viral vector. So we use a harmless virus, one everybody has and has been exposed to multiple times. And into that virus, we take out its DNA and RNA and we put in RNA or DNA. So that would make protein immediately, or DNA that can be transcribed and translated into proteins. That is a part of a microbe. Usually something that we have know that makes many people make an immune response to that piece. Now, a whole cell vaccine is great because like its advantage, like why we still even use these, is because you make the best immune response to this because it's the whole thing, right? Or a very specific targeted piece of it. That means you need fewer booster shots for this, right? But it does mean there's probably going to be some allergic responses in some people. Some people may get from the live attenuated, they may develop a partial infection, they may be sick for a couple days. So they're good and bad, right? Like everything, you know, there's always some risk. Now, where's my other page? 436, where's 437? Okay, now, we can also use pieces of a vaccine. Go back in here. All right, we call those subunit vaccines usually. And in this case, it's only the antigen. And like I said, usually the antigen that is known to make the most people have an immune response for it. So you can isolate, meaning you can try to purify the antigen from the live organism and then kill the organism and then separate it out so you just have the antigen. Okay. Or you can artificially make them with recombinant DNA. So you can take the DNA that makes the message, that makes the protein for that particular antigen, and you can put it in a yeast cell or an E. coli cell or something that will make a lot of that antigen and then get rid of the excess that way. You can just get a vaccine that is that contains DNA or RNA for the microbe. So this is like the COVID-19 vaccine. and you put the DNA and RNA into a lipid vesicle. So that means a bunch of phospholipids mixed with water, they automatically form little spheres. And you can get them to take out the DNA or the RNA, and then you can inject that, or people can inhale it, whatever. And then it will fuse with your membranes, because it's membranes, phospholipid, and dump that DNA or RNA into your cells, and you'll become target cells, right, for cytotoxic T cells, and or you will process that antigen and say, here it is, here's triangle antigen with class 2 MHC, come make an immune response. And the third one is sometimes we call these conjugate vaccines. And I don't know, I think I may have talked about this a little bit the last time when we talked about things that are antigens. And I said that proteins are the best antigens, but polysaccharides if they're really long, right? So a conjugate vaccine would be like the pneumonia vaccine, the Streptococcus pneumoniae vaccine. It is all of these polysaccharides plus some peptides or protein to make it antigenic. Right. Okay, so let's look at this schematic again here. Because it's just a little more cartoony, a little more picture, if you want to look at the pictures instead of the words. If you notice current examples, they give some for each of these. So whole cell virus, the injected flu shot is whole cell virus, attenuated, live attenuated MMR, measles, mumps, rubella, right? That's a live attenuated. There's the COVID-19, the Johnson & Johnson vaccine. That was also a whole organism. They took out, that was like this one, where they took out put it into a different viral vector. It wasn't as effective, especially at first, right? Subunit vaccines or pieces vaccine, the hepatitis vaccine is a subunit vaccine that was probably one of the first ones that was developed. So we're going to use another virus or bacteria to just make the surface antigen, because that's the thing that you make an immune response to. Then we're going to collect up a bunch of those surface antigens and just give you the antigen. You're not actually getting any of the hepatitis virus, but you're not getting any of its machinery, so it can't reproduce in you. You're just getting the antigens so you can make a primary immune response and be ready for if you ever see it. The Pfizer and Moderna COVID vaccine shots were messenger RNA in a lipid vesicle, right? That was huge, like step forward in vaccine production. And then Haemophilus influenza is a conjugated. So that's another respiratory infection. The infants get Haemophilus type B, I think at birth or close, maybe a month or so after they're born, which brings me to this picture, which I've lost already. So here is the recommended immunization schedule, and this is just for children. There's one for adults, like if you've missed doses, or if you're headed to a country or a place where you might get a particular organism, there's schedules for that as well. But this is for infants from birth to 15 months, so that's not quite a year and a half, right? And it's showing the first dose, second dose, and notice the ones that they have to get multiple doses of are usually not the ones that are whole cell, right? Because they're just not as good. Like they don't make as much memory cells, so you have to have them more. We don't keep getting, like you don't get polio over and over again during your lifetime because it's most dangerous to children. And once you've had 3 doses, and polio is only a virus that can infect humans, no other animals. So there are so few cases of wild type polio that chances are as an adult, you're not going to ever run into polio, right? So it's not worth vaccinating you again and again and again for something you're never going to see again. Chickenpox is a little different, right? So is that on this? I don't think did they do that early? Yeah, varicella, there it is at the end, right? At a year, to 15 months. So that one you get then, and then you get again, maybe when you're 11. And then when you're an old person like me, you get a shingles vaccine, which is basically a booster shot for varicella so that I don't get shingles as an old person. Because the last time I had chickenpox, I literally had it and I was 6. So, okay, yes, do I have B cell and T cell memory for that? Maybe. Now it's been 54 years they've been sitting around and probably died, so maybe not. It'd be a good idea for me to get a booster, right? The organization that derives this chart, it's done in conjunction with the CDC, but the organization is called the Advisory Committee on Immunization Practices. And that's, you know, we have to abbreviate that, of course, ACIP. And that committee is made up of people that are pediatricians that belong to the American Academy of Pediatrics and the American Academy of Family Physicians, because a lot of these are things that's who will see children and old people, pretty much, right? In conjunction with the CDC. So remember when we talked about what the CDC monitors or what is a notifiable disease, and I told you a lot of these are diseases we vaccinate against, right? So that's what I mean by in conjunction with. Last week, this committee was disbanded, so we don't have that anymore. So good luck. I find a copy of this before it also disappears, because so far that tends to be the practice. When the committee is erased, the CDC Committee on Sexually Transmitted Diseases was removed, and all the information, all the literature on how sexually transmitted diseases are transmitted was also removed from the CDC website. So luckily you have a microbiology book, so you can do that. And the last thing I want to say is about this, right? Because I think this is confusing to people. We call it herd immunity. And the confusing part about it is it's when we consider that a population, this is when a population enough, or I should say in a population, enough individuals have either, and it is an either, been vaccinated or, right, had the infection, that the population is no longer considered a reservoir. So again, we're using a lot of the words that we learned back in chapter 13, reservoir, too bad I can't spell them, right? Remember that a reservoir is a place where an organism can persist and have opportunity for transmission. So that's what I mean, like about polio and smallpox. Smallpox is considered to be pretty much eradicated from the entire globe. The only remaining stores of smallpox are in military facilities. Okay. Polio is almost eradicated, except for a few small populations that have not been vaccinated. But because many, that's probably on the list of things people are electing not to have as a vaccine. So if you got polio, you know, it travels fecal to oral, you get muscle weakness, you can have to be on an iron lung or some version of that. And it affects skeletal muscles, if I'm not mistaken. So a lot of times it affects people's legs. That's why they can't walk, because that's one of your bigger, larger muscles, right? The iron lung is for the breathing part, right? So this is what people are concerned about with the measles outbreaks that are going on, right? Because we're losing our herd immunity. And so as individuals decide selfishly not to immunize their children, they're putting all of us at risk, because what's happening, and you know this from when we talked about antibiotic resistance, those viruses are mutating and they are still in the population. And everybody who gets those infections is harboring the mutants. And eventually there'll be enough mutated that all of us that have been vaccinated, we're going to start getting these things, right? And I get it. Your measles, what, 3 people died in Texas. So you're like, oh, that's not a big deal, right? The one that's the most frightening for me is rubella, because that's one that when a mother that's pregnant gets that causes deafness in the infant, hands down, no questions asked, no matter what trimester, and usually stillbirth, right? Mom may not even know she has it. She may not get a rash. Dad may have it and give it to mom, and she might not get a rash, right? Just is, right? If you've had the mumps, most of you aren't old enough to have had the mumps. They are most unpleasant. And some mumps versions of the virus have been linked to some forms of cancer. And of course, measles is measles, right? Not only does measles, everyone's like, oh, measles not so bad. Like I said, not that many people died, whatever. What they don't often tell you is one of the side effects for measles when young boys get it is sterility. Because if their fever goes really high, it actually causes them to be sterile or be able to produce less sperm for later in life. And they won't know that until they're in their 20s or whatever, trying to have a family, and they don't make enough sperm, right? So that's for all the Facebook mommies, right? Because it's just so frightening on so many levels. All you have to do is ask your pediatrician. They can space these doses out over 2 years if you want them. They can give them individually. They cannot give them with any other vaccination if you're concerned about the overall effect of more than one vaccine at a time. That's all been worked out by those people that I erased, right? So that's that. We have one more thing, and then we're done. I know I said that was the last thing, but we have one more thing, and then we can go to lab. And those of you that are finished can just hand me your unknown paper and go home. You'll be so happy. Okay. So the last thing is called types of acquired immunity. And we divide it up into, it'll come to me, naturally acquired immunity and artificially acquired. And each of these, we just keep adding words to it. Each of these, we say naturally acquired, you can divide it into active and passive. A naturally acquired passive immune response is usually the strongest. That means you as an individual, were exposed to the microbe, right? You made memory B cells, you made an immune response with memory B cells and memory T cells and lots of immunoglobulin. And so you're immune to that. Our naturally acquired passive is only when you're a fetus, because remember, IgG can cross the placenta. So if mom has a cold, you get those immunoglobulins, right? Or when you're a it's hard to say, neonate, when you're first born, and if you're breastfed, right? You're going to get some IgA from mom in that colostrum those first few days, but that's it, right? So if mom's really sick with something, you're going to get protection from that, which is probably a good thing because you're going to be spending all your time with mom, right? Especially when you're first born. Artificially acquired, we can also divide into active and passive. active is when you are vaccinated against a particular infection and you make an immune response, right? Like I said, it's not as great as having the vaccine, but you're making an immune response, and you might get a booster and another booster, get tetanus. Right? Your response to tetanus doesn't last very long, so you need boosters throughout your life. Or naturally acquired passive. This is pretty rare. This is when you get pooled, we say pooled serum from people that recovered. People, patients. So sometimes this is like if you get a snake bite with a poisonous snake, or like a jellyfish that has the poisonous toxin, or a spider. You live in Australia, right? You might get antitoxin, which is pooled serum from people who've had that infection and lived, and they collect blood from them over time, like for a month, and they suck off all the serum and they save it so that when somebody comes in an emergency, they can like give you some help immediately, right? A lot of times though, you will still have a little bit of an immune reaction to that because it's not yours, right? And they can't purify it from all the cells in the serum, and your body's going to go, wait a minute, that's not self, right? We're not shaking hands, we don't know each other, right? So sometimes it works, but also causes a little bit, we call it serum sickness. Okay, that's the end, I promise. Hey, stop. Yes.